Hello students, this is Faizan Mirza. We are discussing genetic technology and its application in agriculture. So in agriculture, the basic the requirement of incorporating genetic technology is to improve crops, which means that you can modify the crops. The examples include you can get golden rice or canola seed for the oil. You can get herbicide resistant crops, and not just this, you can get insecticide resistant crops as well. So these are the basic three things that we'll be discussing and covering in this particular video or this part of the series of lecture. So let's start with golden rice. So what's golden rice? Golden rice is vitamin A enhanced or vitamin A enriched rice. So a person who is taking golden rice is having a lot of vitamin A going to their diet, and since vitamin A is already there in this golden rice, the person will not need another source of vitamin A. So people who suffer from night blindness and because of malnutrition, so if golden rice is given to them, they won't need vitamin A from any other source. They can just eat rice, and from that rice they can get vitamin A. And vitamin A lack cause night blindness, so if they are given enough vitamin A, so night blindness can be averted. So, how is golden rice produced? So, what do? You take carotene gene, and the carotene gene is collected from the plant maize. This is the first thing. And then you take the bacteria. The bacteria is Agarobacterium tumefaciens. So, this is the bacteria. Again, you can just work out what's the genus and the species of this bacteria. The plasmid is collected from here. You just produce a nick, and a recombinant plasmid is produced by adding the gene of carotene into the plasmid of this Agarobacterium. This makes the Agarobacterium tumefaciens to be a transgenic organism. This will carry the gene, and this will become the actually vector for the carotene gene to be delivered. The character is actually the plasmid in this bacteria. So the vector will carry the carotene gene, and this A tumefaciens, if this is allowed to infect the rice embryo in lab, so the rice embryos, they will receive the bacteria. So infection of this bacteria will not cause any disease because it's a non-infectious bacteria. So you actually allow the bacteria to just penetrate into the embryo of rice seeds. So when they go there, the transgenic rice seeds with the carotene gene is already there in the endosperm. So this is how you get this vitamin A enhanced rice. So when you plant these rice seeds, so they will just grow on to become vitamin A enhanced rice, and this is how golden rice is actually produced by genetic technology. Another thing is you can produce canola seed. Canola seed is the oil seed that we commonly use in oil consumption at our homes. So oil seed is actually coming from the plant called Brassica napus. It is a source of vegetable oil, and it is having two compounds which are not fit for human consumption. The oil seed, which naturally is Brassica napus. So the undesirable products are rucic acid and glucosinolates. Both these are not fit for human consumption, so they must not be there in the seed, but they naturally are there. So how can you make this seed useful for yourself? So canola seeds are made. Canola seeds are Canadian oil seed low acid, and these canola seeds are actually hybrid Brassica napus seeds, in which the rucic acid and glucosinolates have been, the genes of these have been reduced to such a level that they are fit for human consumption now. So canola oil is commonly consumed globally. Herbicide resistant crops. How can you make a crop resistant to herbicides? So you need to understand what herbicides are doing and what are weeds and what is your crop plant. So glyphosate and glufosinate, these are the commonly used herbicides which are used for killing the weeds. And weeds, they compete for the natural resources in the soil with your crop plant. So the crop plant will be having stiff competition from the weeds in the natural resources in that region if you spray the herbicide glyphosate. How glyphosate would function? You spray glyphosate on the crops, so the crops and the weeds, they both get killed. So crops will be affected as well because glyphosate is equally poisonous for crops and weeds. How is glyphosate a poisonous, or how is it killing these? Glyphosate actually inhibits an enzyme which is required for the synthesis of three amino acids in these plants. The three amino acids are PHE, TYR, and TRP, which are phenylalanine, tryptophan, and tyrosine. So these are there. So what do you need to do? You need to just make sure that these glyphosate is sprayed, these enzymes, the enzyme which is required for synthesis of these three amino acids, it gets inhibited. And since this enzyme is inhibited, these three amino acids are not made. If these amino acids are not made, the weed and the crop dies. So how can you make your crop resistant? The crop can be made resistant, so in this way. So what you need to do, you have to incorporate a gene from agrobacterium. Agrobacterium is having an alternate of the same enzyme. This is the same enzyme but a slightly variant version of the same enzyme, which can still synthesize the same three amino acid, and this enzyme is not affected by glyphosate. So if you somehow take out the gene from this agrobacterium and you transfer this gene of this enzyme into your crop plants, so the plant becomes transgenic crop, and this transgenic crop will grow in the soil, and the weeds will also grow together with it. Now if you spray glyphosate, so the crop will survive while the weeds will die. So the crop has been made herbicide resistant by this particular process. So this is how herbicide resistant crops are made, but there are some ecological concerns of using herbicide resistant crops. These mainly include the detrimental effect that they may have on the environment, which include: number one, the genetically modified plant can become an agricultural weed. In fact, it becomes an agricultural weed. The pollen can transfer from the gene to the wild relatives, producing hybrid offsprings that are invasive weeds themselves. And then herbicide resistant weeds will evolve because so much of the same herbicide is used, so they will keep the weed itself will start to evolve. So the glyphosate resistance, how is it actually working? This is the sprayer which you are using to spray this glyphosate. So glyphosate is sprayed on the crop, and this is your crop, and these are the weeds. They both are competing for the same resource in the soil. So what do you do? You take glyphosate spray, you spray it on the leaves. The leaves take these to the inner tissues and of the weeds and the crops. The weeds are killed, and this is the weed, this is the weed, this is the weed. So the weeds will get killed because the enzyme required for synthesis of the three amino acids is blocked. Whereas the transgenic crop, this is the transgenic crop. You can see how happy the transgenic crop is. It grows very well, it grows better, and the better yield means that better it is not having any competition with it for the natural resources, because it has been removed from the environment. So these weeds are killed, their nutrients are returned to the soil, which will benefit the crop even further. So this is how herbicide resistant crops actually work. These are some benefits and the potential dangers of why the glyphosate resistant herbicide crops are useful and how they can be a nuisance. So probably you can just read these yourself, and you can just basically they cover the same 3 points that I have discussed a bit before. So now coming to insecticide resistant plant or insecticide resistant crop. How can you make insecticide resistant crops? So what do? Corn borers are a nuisance for maize, for the crop maize, which actually is corn. So corn borer is an insect that adversely affect cornfields, and cornfield is the maize field, by the way. So what do? Insect resistance gene can be incorporated into maize. So if there is insect resistant gene is entered into maize, so insect resistant maize is produced. What gene is this? This is called as the cry gene. The cry gene codes for Bt toxin. Bt toxin is coming from a bacteria, Bacillus thuringiensis. So this is the bacteria. From this bacteria, you take out the Bt toxin gene. This Bt toxin gene is called as the cry gene, and you incorporate this into the maize. Transgenic maize is produced by transferring the Bt toxin cry gene into the plant maize. The crop can now become insecticide resistant because it is called Bt maize, since it is having the Bt toxin now. Corn borer, if it comes and it tries to eat its way into that maize, the Bt maize, this Bt toxin will be toxic for the corn borer, and the corn borer will die. So the yield of Bt maize increase. So this is what has happened using insecticide resistant crops such as maize. They have been protected from the corn borer, and overall Bt maize is commonly grown all over the globe and consumed as well. Bt toxin gene is non-toxic for human consumption, so and it doesn't affect many species. It is suggested to be quite safe to be used. Ecological concerns of using insecticide-resistant crop, they are obviously there. So, what are these ecological concerns that can be considered regarding insecticide-resistant crop usage? Number one, again, the detrimental effects on the environment which we are discussing. Number one, the evolution of resistance by the insect pests. Yes, because this is what we'll discuss right after, probably on the next slide. The damaging effect on other species and other insects, and the transfer of the added gene to other species of the plant. So here you are damaging effect of the other species, means you are targeting a certain species, for example corn borer, but you end up killing some other organisms who were the non-target organisms. You were never supposed to kill those, and they were not even harming you, but they also die because you have used an insecticide which is already there in the crop. So some non-target species can get affected, and the transfer of the gene to the other plant species can cause the superbugs to form. So what do you do? How insecticide resistance actually is going on? So corn borer dying from Bt toxin, which actually die from eating that maize, Bt maize, they have a dominant allele. So homozygous dominant and heterozygous dominant, once they all die, they are killed from Bt toxin use. So corn borer who have the dominant allele, be it homozygous dominant or heterozygous dominant, they both are killed by Bt toxin. So let's suppose let's take the example of here, and we are using S for the dominant allele susceptible to Bt toxin. So any corn borer which is having this S allele, the dominant allele, who is susceptible to Bt toxin, would be killed by eating the Bt toxin or Bt maize. Corn borers recessive for the same allele, for the same gene, they survive. So homozygous recessive corn borers are found in population naturally. They already have this mutation, and because of that mutation, they are resistant to Bt toxin. So variety of corn borers who are resistant to this, they can be given the letter, for example R, as a recessive allele of resistance to Bt toxin. So corn borer, if you just go for this for the sake of genetic cross, the phenotypes for susceptible to Bt toxin genotype is SS and SR. They both are sensitive to Bt toxin because they have the S allele, which is the dominant allele. So a corn borer who is homozygous dominant or heterozygous dominant for this allele would die by eating or feeding on the Bt maize. But the phenotype of the resistant to Bt toxin will have RR. They are both resistant allele. They are homozygous recessive condition. So this corn borer can just not be killed by using Bt toxin. So what happens? They beat these Bt toxin resistant one, they reproduce, and since this is a recessive allele, so when they reproduce, 100% population of recessive are produced. So this is how corn borers are increasing the population of resistant corn borers who are resistant to Bt maize around the globe. So if they increase their population to a large number, eventually all the corn borers will be resistant to this insecticide resistance Bt toxin gene, and then using this insecticide resistant Bt maize, which is Bt toxin maize, will not be working in our favor anymore. So as you know, this is the recessive condition. So whenever two organisms who are recessive for the same trait, they fuse together or they mate together, the 100% population will be having that recessive trait in the phenotype as well as in the genotype. So 100% resistant corn borers of Bt toxin can be produced. What is the solution? The solution is to go for refuges. Refuges are fields with 50% Bt and 50% non-Bt toxin maize all grown together. So when both of these are grown together, the Bt maize is the one which is actually having the Bt toxin, and non-Bt maize is not having that Bt toxin. So this promotes the survival of Bt toxin vulnerable dominant allele, and it lowers the allele frequency of homozygous recessive Bt toxin from being passed into the next generation. How so? We can just perform the cross, and we can see that if two homozygous dominant for sensitive for Bt toxin are crossed together, you can get this kind of a ratio that all 100% offspring will be of this sort. If two heterozygous dominant are crossed, so again you are passing all four of these will have the dominant allele, so they all are will be, they all these will be 100% susceptible to be killed by bt toxin. So when you plant refuges 50% bt toxin and 50% non bt toxin maize in the same field, so you actually surprise the corn borers. So when you surprise the corn borers, so some of them survive, some of them die. Yes, your yield of bt maize will decrease because the bt maize which is non bt, or you can say the non bt maize will be easily eaten away by all these corn borers. Your yield will decline, but you can delay the allele frequency to increase, which otherwise could play havoc with whatever you have done in the genetic engineering. The question comes at how safe are the transgenic plants. For example, BT maize corn seeds, they disperse up to 60 meters. So what we see, the maize fields, normally wherever maize is growing, there's another plant, milkweed. It grows on the edges of these fields. It doesn't harm the plant, and this milkweed is a food source of caterpillars of monarch butterfly. So monarch butterfly is a non-target organism of yours, and milkweed also is non-target, and you are growing your bt maize in that region. So how can the bt maize affect milkweed and this monarch butterfly? So caterpillar of monarch butterfly, they were fed on three kind of milkweed pollen grains. In an experiment, they were fed on milkweed with no pollen, with actually no pollen maize, and they were fed on milkweed with pollen of non genetically modified maize, and they were fed on milkweed with pollen of genetically modified maize, which is a bt toxin. So this was fed on bt toxin pollen grain, this was fed on the natural pollen grain, and this was kept with no pollen grain from maize at all, to compare that how the caterpillar of monarch butterfly is affected by use of transgenic plants in this way. So in this experiment, 56% of caterpillars survived on feeding pollen from the bt maize. So this include that bt maize, out of these 56% of these survived, 44% of them died. So this was considered safe. Why? Because in this experiment, although this 44% is a good number to say that they probably are not very safe to be used, but in natural environment, caterpillar are not present during the season when the pollination from maize occurs. So when this maize will be pollinating, it will be pushing out its pollen in its environment, which will land on milkweed. So caterpillars will not be there. The monarch butterfly will not be in the season to form caterpillars, and the season in which the monarch butterfly actually form caterpillar, the bt corn seeds will not be producing pollen to land on the milkweed. So this is why it's considered safe. Another way there are other strategies as well to prevent the spread of these pollen grains to long distances. So what needs for to curtail the damage for the ecological concern from the transgenic organisms? So what's done? One very important feature is to have a very good idea of safe distance for the plantation of the transgenic varieties. You should know how safe it is to plant it at a certain distance, so the pollen grains or their seeds do not disperse long distances to affect different organisms, the wild relatives or the weeds in that region. So safe distance must be taken for consideration: the mode of pollination, dispersal, and the distance to which how distant the pollens and the seeds may disperse. One more important way, other feature could be you can use electric fence to prevent the wandering herbivores, to reduce the chances of long distance the dispersal. And how can this be done? This you can see in this figure. These are the electric fence on this crop field. So although these things have been useful, so long-term effects of on health and consumption of these GMOs or these transgenic organisms is not known. And a very important point to consider is that absence of evidence is not the evidence of absence. So calculated risks should be taken, and this is where we end the genetic technology, and this is where we wrap up our topic, and that's it from my side. Thank you so much.